Hey everyone, today we are going to start with chapter 12. Our goals for today are going to be to name the major regions of the adult brain and to name and locate the ventricles of the brain. So this chapter is filled with a lot of really incredible things that our brain can do. It's a little lengthy, it's a little in-depth, but hopefully you learn something and enjoy it along the way. So this is important because understanding the central nervous system contributes to your work with brain and spinal cord injuries such as a stroke. So you may know someone who's had a stroke, or you may at some point in your life encounter someone who has a stroke, and this helps to make some of those things make sense. So the adult brain has four major regions. Our cerebral hemispheres, we have a left and a right. The diencephalon, our brainstem, which also consists of the midbrain, the pons, and the medulla, and also the cerebellum. We're going to start with the cerebral hemispheres before we delve into the other three. So if we were to look at a diagram of what your brain actually looks like, you can see cerebral hemispheres here, kind of the gray outer covering. We have the diencephalon in the middle, the cerebellum just underneath that, and then, cerebral hemisphere here. Diencephalon, our cerebellum is going to be this kind of spaghetti looking guy in the back, and then finally we have our brainstem in green. Okay. So the major regions and the organizations of our brain, we have gray matter and we have white matter. So our gray matter is going to be short, non-myelinated neurons and cell bodies. We already know if something doesn't have myelin, that means that the signal isn't transmitted as quickly. Whereas our white matter contains mostly myelinated, but some non-myelinated axons as well. So the basic pattern that we find the organizations of the gray and the white matter in the central nervous system is generally a central cavity surrounded by gray matter, with the white matter being over top of that or external to that gray matter. We do have some changes of these patterns. The spinal cord is going to exhibit this pattern. However, it's going to have changes as it descends down into the brainstem. Our brainstem has its own additional pattern. The cerebral hemispheres have their own as well. The serum and the cerebellum have different scatterings of gray matter and nuclei amid the white matter, which we're going to dive a little bit deeper into next unit. So if we were to look at these organizations of gray and white matter, so first example right here, our spinal cord. We can see the large intersection of gray matter surrounded by white matter on the outside. So if you look back down to this helpful little key down here, gray matter, non-myelinated, white matter, mostly myelinated. So that is a cross-section of our spinal cord. Now, if we're to look at the brainstem, we have a little bit of a different pattern here. We have an inner gray canal, we have outer white matter, but we also have some gray matter scattered in there as well. Okay, and then finally, so this is coming from the brainstem, and then finally, if we take the cerebrum here and we cut that in half, we're going to be looking at a mostly white matter inner core. We have some gray matter scattered within there as well. Look at it. And then also topped off with some gray matter. So even though we have variations in these patterns, it's typically central white, external gray, obviously with the exception of our spinal cord. And rounding out this chapter, we have our ventricles. So ventricles are going to be these fluid-filled chambers that are all continuous with one another, so they're not separated, they're not in chunks, but are going to be filled with cerebral spinal fluid. And the inner workings of these chambers are going to be filled with ependymal cells, which, if you remember from last section, are neuroglia that have the cilia that help support and circulate the cerebral spinal fluid. So these are essentially hollow chambers with fluid on the inside, and the reason that these are important is they give lightness to our brain. So these empty chambers help to give the brain some support from the inside internally, and nourishment, and also to help lighten the brain. So we have paired ventricles, so one on the left and one on the right. So each of these ventricles is connected to the third ventricle by the interventricular foramen. And we know from previous chapters that foramen means a hole. So in this case, it's like a conduit connecting this third ventricle. Okay? And the third ventricle is going to connect to the fourth by the cerebral aqueduct. So if we take a look at these, this is essentially what they're going to look like inside of our brain. So we can see we have this lateral ventricle, we have our third ventricle here, we have our fourth ventricle down towards the brainstem a little bit farther. So there's the cerebral aqueduct that we were talking about, and then this interventricular foramen, so this little hole that's going to connect the two of them. So again, these are going to be filled with fluid that circulates to help give our brain some nourishment and also support and protection from the inside, and to help give some lightness to our brain. It's essentially helping it float within itself. So it's the end of section 11.1, and we will move on to section 11.2 shortly.